So we were talking about metabolism. We'll be starting with metabolism. And the first main thing is metabolism. We take in the food. In that we have no voice. Okay. Yeah. So in food we have fat, carbohydrate and protein, and all the metabolisms of fat, carbohydrate and protein ultimately lead to formation of pyruvate or acetyl coenzyme A, which feed into the citric acid cycle that produces the NADH and all that, and then that goes into the electron transport chain which produces ATP. Okay, so in today's class we'll start with glycolysis, right? The glucose metabolism part. So where does the glycolysis part takes place? It takes place in the cytosol of the cell. So this is your MCQ. Okay, and at the end we have formation of pyruvate. Okay, it's a 10-step process out of which 3 steps are regulated. Okay, they are kind of irreversible. Okay, so if we have oxygen, then fine, you know, the pyruvate continues to go into the next cycle. But if we don't have oxygen, the pyruvate forms lactate. That's the point over here. Okay, so as we move on, you will understand what we were talking about. I'll come to the overall reaction later. Let's start with the cycle. So we have glucose. All this is taking place in the cytosol of the cells. Okay, it's not inside the mitochondria. It's in the cytoplasm. Right. So we have glucose. You see glucose here. Glucose is converted to glucose 6-phosphate by hexokinase, and we have 1 ATP getting used here. Okay, so when we say hexokinase, now at this particular point in your keynotes, I want you all to make a note of one thing. This hexokinase is, this enzyme is used in all the cells. But in liver, we have a special enzyme that is, you know, kind of, we use the word in the last class, isoenzyme. It's an isoenzyme that is called as glucokinase. So what does an isoenzyme mean? It's mediating the same reaction, but the two enzymes are different enzymes, but they are mediating the same reaction. Okay, so in liver, the same reaction is mediated by glucokinase, but in all the other cells, it is mediated by hexokinase. Okay, hopefully that's clear. Then we have, after glucose 6-phosphate, we have it converted to fructose 6-phosphate. Here we use the isomerase. After that, we have fructose 1,6-biphosphate. Now you see we had used 1 ATP here. Okay, here we have used 1 more ATP. Right, so this is phosphofructokinase 1. Then we get fructose 1,6-bisphosphate, which is broken down. Fructose 1,6-bisphosphonate is a 6-carbon sugar which has the phosphates attached to it. Now we break down the 6-carbon sugar and we break down into 3 each on this side. So we get 3-carbon sugar, that is dihydroxyacetone here, and 3-carbon sugar, that is glyceraldehyde 3-phosphate here. Okay, are you with me till now? So now this dihydroxyacetone phosphate or glyceraldehyde 3-phosphate, they are interchangeable. So you can say we get from glucose 2 molecules of glyceraldehyde 3-phosphate. Yes, they are aldehydes and ketones. Right? So from one glucose molecule, we get 2 molecules of glyceraldehyde 3-phosphate. Isn't it? So after that, whatever reaction continues, whatever we get, it will be kind of a double because we get 2 of those things. Isn't it? So this is the point you need to understand. So till this process, what we did with the ATPs, we used up 2 ATPs here. Okay. And here, there's one thing you have to notice is this reaction, this one, phosphofructokinase 1. This is the rate limiting step. So if you're making a note in your keynotes, please do that. Although it is there in the chart, but if you do it on the flowchart itself, it's easy for you. Dhara, the same DHEP is converted to this. There is an isomerase. They can get converted to one another. So ultimately we get this two of these. Okay, they are interchangeable. Okay, now that glyceraldehyde 3-phosphate, after that we continue over here. At this particular stage, that is glyceraldehyde 3-phosphate dehydrogenase, we get 2-NADH, right? Every time we are considering two molecules here. Look at this, two molecules total. Actually, we are metabolizing one molecule of glucose, but we get two molecules of this. So from here onwards, we are considering it 2 times, everything 2 times. Okay, now look at this here. At this particular step, there is a formation of 2 ATP. See that? This is a phosphoglycerate kinase. After that, we continue here. There's one more thing I want you to notice. You look that here, it's enolase. If you remember, this is the enzyme which is blocked by fluoride. Isn't it? Yes. Okay. After that, you see here, we have again 2 ATPs formed. So, if you remember in the first part of cycle, what we had was 2 ATPs being used up. But in the second part of cycle, we formed 4 ATPs. Isn't it? So, actually, how many ATPs you get in total? 2 we used up, we formed 4. So, how many you got? 2 ATPs. So, net 2 ATPs you could save. Other than that, you produced 2 NADH. Other than those 2 ATPs, you produce 2 NADH. You will be using these 2 NADH, in the electron transport chain to produce more ATPs. Right? So, hope this is clear till now. I'll be talking about ATP's calculations as we move on. But at this point, I want you to make a note of 3 regulatory steps that we had been talking about. I told you, right, there are total 10 steps, out of which there are 3 regulatory steps. The first regulatory step is this one, the hexokinase. The second regulatory step is phosphofructokinase. Okay. And the third regulatory step is the last one, that is pyruvate kinase. Okay, so these are the 3 regulatory steps. The hexokinase, the phosphofructokinase, and the pyruvate kinase. So these are the 3 irreversible regulatory steps. But out of which the rate limiting step is phosphofructokinase. So let me write down here. Regulation at 3 points. Regulation at 3 points. One is hexokinase. Second is phosphofructokinase 1. The third is pyruvate kinase. Okay, just try to remember it today here itself. These are the 3 regulatory steps, out of which phosphofructokinase is rate limiting step. Okay. There's one more thing which you need to know. Remember, I told you that we have used up 2 ATPs. We have used up 2 ATPs and we formed 4 ATPs directly. Okay, other than that, yes, we did form 2 NADH, but we'll be using NADH later in the electron transport chain to form ATP. Right now, we don't have ready-made ATP from it, isn't it? So, what are the ready-made ATPs? We made 4 ready-made ATPs and we used 2 ready-made ATPs. These using up and making is called as substrate level phosphorylation. Okay. Substrate level phosphorylation. Okay, so in substrate level phosphorylation, we formed 4 ATP, we used 2 ATP, and net we got 2 ATP. So you understand the significance of the word net here? It's a total. Yes. So at substrate level phosphorylation in glycolysis, we get net 2 ATPs. But total we generate 4 ATPs and we use 2 ATPs. Okay, so read your questions very carefully when you actually come across substrate level phosphorylation. Okay, but now you look at this NADH. Okay, now this is, this chart actually tells you about whatever I told you during the process. Okay, so I'm gonna skip through this. This is for you to use it for your reading. Okay, I told you just now, Saurabh, right? Whatever is direct ready-made ATP made during phosphorylation, whatever is made direct, right? See, we are making ATPs and we are also making NADH. Isn't it? So this NADH will be using later in electron transport chain to make ATP. But right now, what are the ready-made ATPs we made? We made 4 of the ready-made ATPs here. These ready-made ATPs are substrate level phosphorylation. Okay, so we made 4 ready-made ATPs and we used 2. You don't have to remember each and every molecular name here, but there could be simple questions. There could be simple questions about, what converts glucose 6-phosphate to fructose 6-phosphate. Now this is an isomerase. Now this is, you just stick on to whatever is asked in MCQs. I don't say remember all the steps. But anytime you get an MCQ, don't try to mug up the MCQ. Rather than that, come back to the chart and see which step the MCQ is about. Okay. And then look into the step. Automatically, once you solve the MCQ, you will realize that you are familiar with all the significant part of the cycle. Right? So don't try to mug up the MCQ. Please come back to the chart every time you get an MCQ. Right? So that will do the job. That will actually filter out what you need to read and what you don't need to read and all that. It will just do the job that way. Okay? So you understood what I want you to remember at this particular moment. I want you to remember that we have total 10 steps. In the total 10 steps, we have 3 regulatory steps. Hexokinase, phosphofructokinase and pyruvate kinase. Out of which phosphofructokinase 1 is a rate limiting step. Right? Yes. Other than that, I discussed that we are forming 4 ATPs at substrate level phosphorylation. We are using 2 ATPs from substrate level phosphorylation. So we are getting net 2 ATPs at substrate level phosphorylation. Other than that, we are getting 2 molecules of NADH2. One second, guys. Yeah, we are getting 2 molecules of this NADH, which will be putting it into electron transport chain. Every 1 molecule of NADH will give us 3 molecules of ATP. Okay, so we had total 2 molecules of NADH. So we'll get 6 from the electron transport chain. You get that point? Already we had saved 2 from substrate level phosphorylation. We get 6 more. So 2 plus 6 total we get 8. 8 saved ATP. Is that clear? We are going to see this calculation again as we move on. But if it is clear right now, we'll move on next. Okay. So in this chart, I'll just move on directly because this is all we discussed. Okay. So under aerobic condition, what happens is, at the end of glycolysis, we have pyruvate. Right? If the conditions are aerobic, this pyruvate is taken up by mitochondria. Okay, after conversion to acetyl coenzyme A. So it's not the pyruvate that enters mitochondria, it is converted to acetyl coenzyme A, and acetyl coenzyme A goes inside the mitochondria. Once it goes inside the mitochondria, we saw there, right, that it goes through citric acid cycle and it is oxidized to CO2. At the end, at the end of the citric acid cycle, we get CO2. That is what is released in the cells, isn't it? Okay. And other than that, we also get a lot of NADH and all those things which we'll be using up to make more ATP. Okay. So, that is what will be the citric acid cycle. But if the conditions are anaerobic, what happens is, this NADH which we had made through glycolysis, which was supposed to go to electron transport chain, now that is prevented. Get my point? So what happens now? That is not happening. Now the pyruvate that we had actually got from at the end of the glycolysis, that is converted to lactate. The enzyme is used as lactate dehydrogenase. Okay? And this particular NADH, okay, it is the one that is converted back to NAD plus during this step. Okay, so during pyruvate getting converted to lactate, let me show you here. Pyruvate is getting reduced to lactate. That's what I'm talking, Radhika. This is happening in anaerobic condition. In anaerobic condition, NADH is not taken up by mitochondria. It doesn't go to mitochondria in anaerobic condition. For that, we need aerobic condition. Okay, so that's what is the normal aerobic condition. But in anaerobic condition, this is not happening. What's happening here is the pyruvate that we had actually received now gets converted to lactate. Okay, for that we use the enzyme lactate dehydrogenase. Okay, and at this step, your NADH is converted to NAD. Because this H is now taken up for the conversion of, no, here the acetyl, it's not going to the citric acid cycle. It's not. Nothing is happening. No mitochondria role because there is no oxygen. Mitochondria can't work without oxygen. We don't have any oxygen. This is a step. This is an anaerobic metabolism of this thing. So remember the two NADH from which we were supposed to get 6 more ATPs. Now we have used them up. And we have actually got what is lactate. We don't get those extra 6 ATPs. So what is the number of ATPs we are going to get if there is no oxygen? How much is the remaining ATPs? Only 2. Because that NADH which was supposed to give us 6 more ATPs is now being used up in conversion to lactate. One more thing that is happening when the conditions are anaerobic is that there is a formation of lactate. Okay guys, is there any delay or any this thing? Actually, my system's hung. I'll give a quick refresh and I'll come back. Hopefully you're clear with this part. All clear till now, right? So what is happening in anaerobic condition is we are getting lactate. We are using this NADH up to make NAD. Now see the plus point. See the plus point here. You don't see any plus point? I tell you the plus point. We are getting back that NAD. In spite of having no oxygen, we can use this NAD again in glycolysis. Remember, we need NAD in glycolysis. I'll show you that. See this? We need form NADH. If we don't have NAD, this step will not go at all. This is a coenzyme, isn't it? So if we don't have NAD, our glycolysis will stop. So in absence, normally where do we get these NAD? We actually, what happens is our NADH which is formed here, this NADH goes into the TCA and all that, this NADH goes into the electron transport chain, and from there the NAD is formed back, and it comes back to be used into glycolysis. But now in the lack of oxygen, our electron transport chain is not working. Electron transport chain is not working. We need this NAD so that at least our glycolysis is working. Now, for glycolysis to be working, we need this NAD, and this is formed back in the next step here. In the absence of oxygen, we have lactate, and here the NAD is formed back. Okay, it uses back again here. Yes, in aerobic condition, it stops at pyruvate, you can say. After that, we consider it, the entry into TCA. So TCA actually starts from pyruvate. Okay. So in anaerobic condition, it continues to form lactate, and the accumulation of lactate that we see in the muscles, this is it. Clear? So this is again what we have already saw. The reaction is catalyzed by hexokinase, the regulation. Okay, these are the major site of regulation of the glycolysis. They are the irreversible steps, and out of which your phosphofructokinase is a rate limiting step. Yes, you can consider them same. Right now you can consider them same because lactate is the one that gives rise to the lactic acid accumulation, we call it. Okay, in this chart, what we actually got, if you remember, see that at phosphorylation at substrate level, we got 2 phosphoglycerate kinase. At pyruvate kinase step, we got 2. If you go back and look at the chart, you will know ATP at substrate level. Okay, and we used up 2 here. Okay. Now look at this glyceraldehyde 3-phosphate. Here we got NADH. NADH is the one that is giving us 6 ATPs. Okay, so now you know that actually if we don't consider our using up, we actually are forming 10 ATPs, but we use up 2, so net 8. So please understand the significance of the word net. Okay, again at substrate level, actually we are forming 4, using 2. But net, we are forming 2 ATPs at substrate level. So you're clear with what we did with the glycolysis?